we should, I guess then we should get going. So thank you all for coming. And today we've got two great talks planned. Just before that, though, just to reiterate, as it says on here, that this speaker series is leading up to culminating in a virtual conference, SynCell 2021. And as it says on the slide here, registration for that virtual conference is now open and is free to register. So if you go to the link at the top of the slide there, you can register for the virtual conference that will be on May 18th to 20th. So I think without any further ado, we should get started with the first talk, which is going to be given by Professor Andy Ellington. Professor Ellington is a professor in molecular biosciences at UT Austin. He is the Nancy Lee and Perry R. Bass Regents Chair in Molecular Biology and the Wilson M. and Catherine Fraser Research Professor in Biochemistry. And he's also a member of the Center for Systems and Synthetic Biology. His lab is working on developing novel synthetic organisms, and they do this by altering the translation apparatus, and they also work on modular nucleic acid software, with various applications, including point of care diagnostics, which I believe will be the title of his talk for today, and also for other potential applications in cell-to-cell communication and drug delivery. Professor Ellington has been a leading figure in fields of synthetic biology and molecular computing for many years, among others. So I'm very much looking forward to his talk. So Professor Ellington, do you want to go ahead and share your slides and begin when you're ready?
There we go. I think that did it all. Can you see my screen?
Yes.
All right, very good. So today, yes, I'm going to talk about cell-free operating systems for diagnostics. That's going to mean there's more in vitro and less cell, although I'll emphasize the overlap. For me, it really starts with how we're developing isothermal amplification assays for point of care assays, as we just heard. Thank you very much for the very nice introduction, Pat. And then something about sample preparation, and that's where cells will come in. And finally, some recent advances that haven't really been published yet. And as a background theme, I would like to emphasize how working in cell-free systems gives you exceptional control over what you can attempt. So no matter what you're doing, removing the complexity of a cell really helps, which is one of the reasons I think so many people are part of this consortia to begin with. With that, I'm going to start in a slightly odd place, which is with advances in nucleic acid computation. And these advances were engendered by, amongst others, Eric Winfrey, Niles Pierce, and Peng Yin. But these were really some of the first founders of nucleic acid computation, at least this generation of nucleic acid computation. And they basically started me off thinking about what I like to call hybridization logic. The ability of G to pair with C and A to pair with T, and how that can be used for programming and computation at the level of base pairing, not at the level of how it necessarily plays out in transcription and translation, but at the level of base pairing. And I would suggest as I go along that this is an important element of cell-free systems, which don't necessarily have to rely exclusively on the types of computations that cells themselves do. In this regard, I'm going to first apply some of this hybridization logic to point of care diagnostics. And our platform there is loop-mediated isothermal amplification, so-called LAMP, which is shown there on the left. Now, in the bottom, just next to LAMP, is a typical LAMP reaction as run out on a gel. And in red is what happens when you run a LAMP reaction out on a gel when you have zero input template. What happens is you get non-specific amplification. The primers will make almost anything. And so therefore, much like a TACMAN probe for PCR, it was very important to develop a probe that could specifically respond to specific amplicons. And that's what you see there in the middle. It is a very simple strand displacement probe that's a hemiduplex, where a single-stranded region of that hemiduplex can interact with the amplicon, in particular the loop of the loop-mediated isothermal amplification amplicon, shown there in gold, and initiate a strand exchange reaction that will eventually split apart, for example, fluorine quencher. But this is the hybridization logic I was speaking of. Pretty simple hybridization logic, at least so far, but very important for detection. Because when we take that gel that has all sorts of muck on it, and we add in this probe, the zero template reaction drops out because it didn't produce anything of great use, and we can detect down to 20 copies of what that reaction was programmed with. In addition, that process of strand displacement, strand exchange, allows you to set up a variety of logical amongst other reactions, and I'll tell you something about those as we go along, and also allows you to adapt these technologies to a wide variety of readouts, which I'll also tell you about as we go along. But again, in the main, and especially for the cell-free aspects of what I'm talking about, being able to use base pairing to make machines, to make logic gates, to make transducers is very important, and again, is something that can be uniquely used in a cell-free setting. Another advantage of this type of probe is that it's exquisitely sensitive to mismatches. And so here we have the detection of a melanoma biomarker where we're either detecting on the right there, the wild type gene, or on the left, the wild type gene, or on the right, a particular SNP. And as you can see in red, we've placed a potential mismatch in the single strand region. And that means we either get complete detection of the wild type with one probe or complete detection of the mismatch of the SNP with another probe. So again, I just wanted to demonstrate that hybridization logic is not only very useful, but can be very, very accurate. In addition, in terms of the outputs, this is an output I'm not going to talk about, but it demonstrates the overall utility of what can be done with hybridization logic. Here, instead of displacing a fluor from a quencher, we displace an invertase labeled with an oligonucleotide from a solid support. So the amplicon gets made, it displaces invertase, invertase goes downstream, cleaves sucrose into glucose, and that can be read by an off-the-shelf glucometer. And so we can readily adapt this type of amplification assay and the accompanying molecular logic to outputs such as glucometry. And on the right there is just shown how you can actually, again, do detection, mainly yes-no detection, of particular templates using a glucometer as an output. Okay, now over the last year, we, like pretty much everyone else, have been working on SARS-CoV-2. So I'm going to give you a summary of some of our SARS-CoV-2 work quickly. As you might imagine, what we did was we developed primer sets and probes that allowed us to detect SARS-CoV-2 RNA. And on the right there, you can see that we can detect relatively small numbers of the SARS-CoV-2 genome relative to a background of human DNA, and give a nice little fluorescent readout in a tube. Now, in part, this is because that we can engender molecular logic. On the left, we actually have an OR gate. What that really means is we have two probes, either one of which will light up in the presence of amplification of one or the other gene. It provides surety for the amplification. And this is not unlike a qPCR test where you'll often amplify multiple genes in parallel, but we can also do this for LAMP. In addition, we can, unlike qPCR, make an AND gate. And this is an AND gated probe that shows the presence of two different amplicons together, where only in the presence of those two different amplicons do we form a bridge between a capture element, fluorescein, and a detection element, a gold nanoparticle, and then can detect that in the context of a lateral flow strip. And this is a particularly useful instantiation of these technologies for resource poor settings that may not have access to higher priced, certainly fluorescence readers, but nonetheless gives you an immediate sort of point of care, point of need readout. But again, both of those examples are based upon the underlying molecular logic of being able to do hybridization and making either an OR or an AND gate for the amplicons, which in turn displace and create these gates. Now, I'm going to get into some of the nitty-gritty of what we've done over the last year. We're able to take this assay and make it work down to relatively low copies of virus in human saliva. And in fact, these are similar to the assays that I can do in my own lab. We have an IRB in place so that we can essentially test our cells as we wish to. And it's not as sensitive, clearly, as qPCR, which will get you down below 10 copies. But it also requires absolutely no RNA prep. These are raw samples where the very fact that we incubate the samples in the presence of the lamp reaction at 65 degrees allows enough RNA to escape from the virions that it can be readily detected. And we published this early on in bioRxiv, and I believe it's now coming out in MSphere. We've also worked with a variety of companies to get this into the marketplace. So this is just the device that, again, I can actually use in my own lab from a local company called Fabrico. It's the Gene Tiger, and will give you, as a small fluorescence reader, an upload of your status based upon essentially not doing nasopharyngeal scrapings, as I show there on the left, but actually at this point, just doing saliva-based testing. We've also licensed some of these technologies to additional companies who are somewhat larger than Fabrico and may eventually introduce them to the marketplace. We'll see how that continues to play out. So that's just an idea of what the system is. Loop-mediated isothermal amplification coupled with probes, coupled with molecular logic based on those probes. But now I'm going to talk a little about why I think this is a particularly good assay, and why, again, in a cell-free context, we can do sample preparation and do point-of-care assays in a very simplistic way. I'm going to give you a couple of examples. And again, these are just going to play upon the fact that we can make primers and probes for virtually anything. Here, what we're doing is we're testing water. And literally, I have groups of undergraduates that go down to the local creek and test for fecal contamination. They look for a particular Bacteroides species. Actually, there are different Bacteroides species depending upon the source of the feces, so we can distinguish human from dog from other animals' feces at this point. But all we do is we draw a bit of water up into a syringe and then push it through a filter, reverse the flow to essentially concentrate the bacteria, and then directly dump it in the lamp reaction, do the amplification, and detect the presence of the Bacteroides. And you can see in a variety of water quality settings, we can irrefutably detect the Bacteroides. And we can do this down to relatively low copy numbers. In fact, one of our great examples is that the students found a burst pipe at the university by looking downstream and realizing there was a huge Bacteroides contamination in the water, which they then reported. Here's a slightly more complex example, and this really does play off of the molecular logic issues that I've talked about before. What we're doing here is we're detecting a wide variety of Zika viruses in the presence of crushed mosquitoes. So up there in the upper right is our preparation procedure. Put mosquito in tube, crush mosquito, resuspend mosquito guts, put it in the reaction. And again, we get nice green reactions when we do this. But because of the way we've designed the primers and the probes, we can both capture a wide variety of Zika samples. Now that's shown there in the middle on the left, where we're capturing genomic variants, both Asian and African, which are not necessarily that closely related to one another, relative to non-cognate viruses such as dengue virus, which is related, but not as close. Up in the upper left are basically OR-gated ways in which we can take all the different amplicons that arose from the different Asian and African species and integrate upwards into a signal. So that eventually at the very bottom there, you see that we are detecting either African or Asian Zika samples relative to controls quite accurately. On the lower right is our positivity rate relative to qPCR from mosquitoes. And again, this is an example of where we were able to use hybridization logic, use molecular logic, use the fact that we were working in an in vitro cell-free setting to garner the results that we wanted. Now, just one more example of this, and then I'll move on. We also have very good detection for tick-borne diseases. And I should have said in the previous slide, we can detect both the disease and the species, the mosquito species in which it was contained. We sometimes have AND-gated probes to say, you have detected this mosquito and that Zika variant. Here we would want to say you have detected this tick and that bacterial disease, Lyme disease. But again, it's the same sort of thing. I'm not making it up. We crush the tick in a tube. We take tick guts. We put them in the reaction. And in this case, potentially for hikers, we're going to run it up a lateral flow assay so you can directly test whether that tick you found on yourself was of a particular species, as shown here, and in addition could contain a particular disease like Lyme disease. This is just results from actual field-caught ticks showing that we can detect their species relative to non-cognate ticks, to ticks that aren't of the correct species to contain the relevant disease state. Okay, so now, having convinced you, I hope, that we have incredibly robust assays, we now want to make our reagents robust. And this is sort of the cell-free aspect of this talk, where we found that essentially we don't even need to make lysates. We can just take cells and either lyophilize them, or as you'll see in a moment, freeze dry them or desiccate them, and they themselves become reagents. So if we have a strain that overexpresses, say, Taq polymerase, it is as though it were Taq polymerase. And that's very useful. And you can imagine in a low resource setting where you don't necessarily have a lot of money for reagents, or you don't want them to sit on a loading dock for 2 weeks in the hot sun and die, that you can make your own. As long as you have bacteria and glucose, you can essentially make your own high-priced reagents. These are just some results from more recent work that, again, is, I think, coming out in PLOS One, where instead of using lyophilization, we actually just desiccate. And again, we can desiccate down a variety of different enzyme reagents. Shown in the middle there is desiccated Taq. That dumped as desiccated bacteria into a tube gives perfectly decent answers. Over there on the far right is desiccated MLV used in an RT-PCR reaction, MLV reverse transcriptase, where again, we can turn RNA to DNA and DNA into qPCR amplicons, dumping in bacteria. And that's probably not going to seem right to most of you, who will be saying in the back of your minds, oh my gosh, aren't you dumping in all the bacterial DNA? Aren't you dumping in all the bacterial contaminants? Aren't you dumping in all the other bacterial proteins? And the answer is yes, yes we are. But they don't affect the ability of Taq polymerase or MLV reverse transcriptase to find the right amplicon, amplify it, and give results that are as good as with a purified reagent. And this is just some of our work in the field. We have amazing partners in Ghana and Cameroon who've now developed their own desiccation abilities and have shown us that they can make their own reagents and use them. We no longer need to send them anything like a reagent. We just send them a single cell with a single plasmid, and they take it from there. And you can see there in the upper right-hand corner, once you make a desiccated reagent, it's good for many, many months, 7 months. Following desiccation, we're still seeing very, very good signal relative to controls. We actually really like this format. We're sort of tired of moving liquid around. And so our scheme for the future is to make desiccated bacterial reagents, put them on discs and handle the discs. And so our future looks something like hole punchers with different reagents where literally you would mix your reagents, potentially buffers freeze-dried onto a piece of paper. You just mix a bunch of pieces of paper in a tube, hydrate it, and do the reaction. And so we see a future where there's much less liquid handling and much more solid handling. And you can imagine for your own labs, potentially the advantages of that. But there are definitely advantages in labs in the field or labs in resource-poor settings for this paradigm. All right, so we've also adapted this to SARS-CoV-2 testing, just like everything else. And this just shows how we are now in the process of making freeze-dried, desiccated, or lyophilized reagents that can be used for SARS testing in the context of lateral flow assays. And again, you see a very good correspondence between what we used to have to do with looking at fluorescence, and even that was pretty simple. We could look on a cell phone with an orange filter and see that, but for folks who don't even have a cell phone, or for whom that option is not a good one, we can do lateral flow assays down to reasonably good sensitivities with cellular reagents. We've made this about as simple as we possibly can. You can make your own reagents, you can make your own reactions, you can do your own analysis, and you can basically make diagnostics that work in the field. Okay, so with that, let me move to further advances for the lab. I think I'm pretty much on time, and I'd really like to save up some time for the next talk, which is going to be amazing, just for those of you who are here. One of the things we've done over the last year is I said at the outset, I said one of the sub-themes here was, wow, if you have control over cell-free reactions, that is true control, that is true power. You can manipulate any aspect of the reaction in a way you just can't with the cell. Well, one of the things we've done is we've begun to manipulate the enzyme. And so while it's nice that there are thermostable polymerases, and while it's nice that they can be put into cells and lyophilized or desiccated and still work, they're not necessarily the best enzymes. And so there's a single DNA polymerase that's used for almost all LAMP reactions. It's called BST, the Bacillus staurothemophilus DNA polymerase. And it is used because it's an incredible strand displacing DNA polymerase. There are very few other polymerases that are moderately thermotolerant and have that property. The only thing even closest is 529 polymerase, and anybody who's ever used that knows they don't want to use that. so we endeavored to improve upon this enzyme. And the first thing we did was we added a folding domain to stabilize it, shown there in the middle. It's called HP47. And it's from the literature, but it hasn't previously been used to stabilize proteins. That made the enzyme that we call BR512. And BR512 is actually as good as BST 2.0, the best enzyme from New England Biolabs, for LAMP reactions. And in fact, it is somewhat better when combined with a stabilized reverse transcriptase, which we call VG125. And so already we've added to the repertoire of enzymes available for LAMP. There used to be really sort of one, and now we would argue that ours is just as good. And that work has just come out. I think it's on bioRxiv, and we've submitted it for publication. But the other thing we did was we used a variety of approaches to stabilize that enzyme. So this is actually protein engineering at its finest, but again, in the context of cell-free reactions, is meant to show you that with control over the system, you can change a bunch of things. We have a very good machine learning portion of my group who went through and using an algorithm that they developed, picked mutations in BST DNA polymerase that they predicted would stabilize it. And lo and behold, the top predictions did stabilize it. You can see there when we heat challenge the polymerase at 75 or 80 degrees, and the wild type essentially drops out, and that's the wild type BR512. That's already improved. We're improving the improvement, and we now get stabilization up to 80 degrees and beyond. Similarly, we took the fusion domain and we put additional positive charges on it, much like the fusion domain of fusion Taq polymerase, where the idea is to give it better contacts with the DNA, and also such supercharging, such additional of excess charge can also in its own right be stabilizing. And once again, we were able to stabilize to heat challenge. And then we just took everything together. We took the best polymerase with the fusion domain, the best machine predicted mutations, the best supercharging, and we put them all together. And suddenly now we can run LAMP, which normally is run at 65 degrees, at 74 degrees. And not only can we run it, we can run it really fast. And so we can routinely get positive answers, positive being the presence of a template, down below 15 minutes. And so we're continuing to work on the development of these enzymes. Again, this work just came out, but we're also happy to work with others on the use of these engineered enzymes, which have been made fairly painstakingly over the past year, but really do the job. In addition, we've been working with the chemistry of LAMP itself. Now, this is going to go in the other direction. Say you don't want to run LAMP at high temperature. Say you are in a point of care setting, and actually it would be worthwhile for you to perhaps be able to put a tube under your arm and run things at 37 degrees. And again, we always try and consider what is the lowest common denominator in terms of doing diagnostics. In this regard, I think, I always like to think of us a little like Intel, right? Intel, you always see the commercials for Intel Inside, the circuitry inside on some device that looks incredibly useful and simple. Well, we're sort of the Intel Inside for molecules. We make molecules that just work, and we don't pretty much care which device they're in, they just work. And again, going with the cell-free mantra, it's really much easier because we could never do this stuff if we were trapped in a cell. In this particular case, we've modified the LAMP reaction itself so that the primers contain phosphorothioates. Phosphorothioates don't bind as well to normal DNA as normal DNA does. They destabilize helices. What that allows is fold-back structures, some shown there in the lower right, that can more easily allow LAMP to occur at lower temperatures. They probably also allow LAMP to occur at higher temperatures. We're still working on the intersection between high temperature LAMP and phosphorothioate addition, but this addition of chemistry allows the reaction to go better. This was only possible because we actually did the hard work of figuring out how phosphorothioates affected the melting temperatures of DNA. We actually went through and did Van Hoeft analysis, over a variety of primers with a variety of phosphorothioates to see how they impacted hybridization and fold back. And this was all work by an incredible graduate student, Stella Wong, who's just about to come on the market for those of you looking for postdocs. Stella is really quite good at both computation and biology. These are just some of our reactions with phosphothioate lamp, so-called PS lamp, where we can both detect down to low copy numbers. On the left there, we're down to 42 degrees, but we're pretty sure we're going to be on the way to 37. And on the right, once again, we can do this with multiplex configurations so that we can detect gene A or gene B and have a more sensitive assay, therefore. Two final innovations, they're decoupled from what I just showed you. This is just a little bit of a grab bag at the end. We're now able to detect methylated DNA in a manner similar to what's been done for methylated qPCR, where you can cleave methylated amplicons and thereby take them out of a population and thereby detect when things have been methylated. And we've been doing this in the context of our GeneTiger devices. So it's even possible into the future we'll be able to do low-end methylation detection for DNA. And finally, here's the real future. This is where we're going. It's been a lot of fun to do fluorescence. It's been a lot of fun to do lateral flow. I don't believe either one of those are the future. I believe the future is electrochemistry, and we are adapting our LAMP assays and our strand displacement probes to electrochemical detections, which I think will allow us to multiplex up to very high levels and be able to make miniaturized devices where, once again, the dare I say it, brilliance of the molecular devices themselves, the intel inside, will play out as nickel-sized devices that can do detection, hopefully in the field. And if those nickel-sized devices need to be made by folks who are working under resource-poor settings, they can go back and do so via the bacteria that we've engineered and via some of the chemistries we developed. With that, I'm at a conclusion, and I just wanted to thank Sanchita Badra, shown here, who literally did almost everything. I talked to Sanchita this morning. I said, here's your talk that I'm giving, which I'm sure is a familiar experience for many advisors, and made sure it had her imprimatur. She was assisted by a very able enzyme engineering team that brought along the recent generations of the BST DNA polymerase. The phosphorothioate modifications, I've already called out Stella Wong. And for electrochemistry, there's been a series of electrochemists in my group, culminating with Dave Walker, who's now doing the device adaptation and miniaturization. And I thank a variety of agencies for support, and we continue to move this into a corporate sector so that it actually can have impact. And with that, I'm open for questions, but again, I'm very much looking forward to Dr. Beng's talk.
Brilliant, thank you very much, Professor Ellington. We've got a couple of questions in the Q&A, so if there are any more, go ahead and type those. I'll read them out, I guess, in the order that they were received. So the first one is from Peter Carr. He says, for POC lamp assays, how is temperature controlled? Is it a standard heating block, thermocycler, handheld heater, or something else?
Peter, that's an excellent question, as usual. So while I've tried to emphasize that we would like to be able to work in real backup beyond resource poor point of care settings, you still need a heat block. At least for the lamp assays that we are currently working with in Africa, you need a heat block of some sort. Now, we've also shown ourselves that you can use a hot glove similar to those that you've used for camping. You can literally just hold the tube in your hand. Therefore, chemical heating is a possibility, and there's some pretty good chemical heaters out there that could be adapted to this. We haven't done that yet. Ultimately, I think we'd like to potentially, again, go to the situation where you can provide your own modest heat on demand in the form of 37 degrees or near there. But right now, you're still going to need a heat block. That is still a requirement that potentially involves an infrastructure challenge.
Question 2 from Irene Chen, which is very exciting. Number 1, it was a 2-part question. Number 1, what information was used to train the machine to predict folding mutations for the BST enzyme? And number 2, what are the next steps and obstacles between your diagnostic technology and hospitals, schools, or the public being able to buy the tests?
Oh my gosh, I could give an entire another talk, Dr. Chen, Irene. No, let me be brief. I'm really excited about the machine learning algorithm. It is based upon an algorithm we adapted from elsewhere, but greatly improved. What it does is essentially looks through the entire protein database, and looks at the pockets for all the amino acids in every protein. And I mean that. And so, for example, we discern the leucineness of an amino acid. So we say, hey, leucine, do you like where you are? And sometimes the pocket will say, yes, leucine is the thing I would most like to have here. And sometimes it says, no, actually, because you've assessed the leucineness and the isoleucineness and the glycineness and so forth of all pockets, I am a sort of pocket that would like to be isoleucine. And so we just look through at any given protein that we have the structure for, and we assess all of its pockets, and we see relative to the database as a whole, whether we can predict more amino acid likeness for a given pocket. That has proven surprisingly successful. What I just showed you is about our 5th example of that. The thing that's not out yet is we have greatly stabilized plastic-eating enzymes and actually can do full-scale plastic degradation with machine learning mutations. So I'm really excited about this algorithm. The algorithm's out in ACS synthetic biology, but the outcomes of the algorithm are going to flow out over time here pretty quickly. The second question is, what next? Well, okay, when we all got hit with this, everybody ran around and said, what are we going to do? And I had assays ready to go, but the problem was, in the absence of an EUA, FDA approval, and so forth, what am I going to be able to do? So that's why I licensed to companies. I licensed to companies so that my work can get out into some sector where it might prove useful. And I think that that's on me that I didn't, say, start my own company or try to work more closely with RADx or what have you. But I've just found that we're best at innovation, and then we license to try and get scaling. With respect to work in point of care settings, we've worked in the past with the Gates Foundation, and we're always happy to work with local entrepreneurs as necessary, because we try and make it, in my view, so that we have a diagnostics revolution where it costs $0 to do diagnostics, and anybody who wants to can. And so that's why sometimes companies are a little uncomfortable with my stance, because, honestly, I'm happy for people to make money, but I'm also happy for people to just do the right thing. Thank you, Irene. Those are great questions. I'd be happy to talk more with you about them.
There's a few more, so I guess we'll try and. Next question from Annibal says, well, basically, what are the drawbacks of the 529 enzyme that you alluded to, and could machine learning improve that as well?
Oh, that's a good question. So 529 is this amazing polymerase that will go forever. It is the basis of some long-read sequencing technologies. It's a phage polymerase. It looks nothing like other phage polymerases. Other phage polymerases do not touch it in terms of its processivity. But it's a mesophilic polymerase. It only works at low temperatures. We have begun to try and improve it. Others have tried to improve it. It is a gnarly little beast. It is great for single molecule sequencing, but it is recalcitrant to a lot of modification. Actually, that's not entirely true. It has been modified extensively, but I'm not sure there are even patents on that. This is a closely guarded trade secret, not from my lab, as to how to modify 529. So far, 529 has been one of the few things we haven't been able to improve by machine learning, but I think we're going to get there. It's just a really unique enzyme.
Okay, and a few more very quickly. Can you get enough primer specificity at 37C?
I'm sorry, I didn't understand.
Can you get enough primer specificity at 37?
I think we can with phosphorothioates, because by destabilizing the helix, we correspondingly say, now thou need more pairing, better pairing in order to work. And so it's a trade-off. The specificity and temperature are always trade-offs, and really it's a specificity stability temperature trade-off. So we're able to dial both directions. We can go up, we can go down, and I think we can match what we need to for primer specificity at almost any temperature.
Okay, next question from Alasina Pasrafshan. For bacteria containing expressing polymerases to be used as reagents, does variability of expression essentially affect the readout? And
No.
how to control?
Surprisingly, no. And one reason for this is, if you think about how much enzyme you add to a qPCR reaction, for example, it's an amount, right? You're putting in 2 nanomoles or whatever. Who set that amount? Where'd that come from? Mostly we just top reagents out. We actually add excess reagents to almost everything we do as molecular biologists. And the overexpression constructs that we have, if added at even a small number of cells, are more than enough. That's really what you need for almost all reactions, is more than enough. And we can easily make more than enough, which is why our results are similar to, or identical to in many cases, qPCR or LAMP reactions run with purified reagents. We have more than enough, and as long as we stay in that regime, everything works fine.
Okay. 2 more questions here. One from Peter Davenport. How does the sensitivity of the electrochemical detection methods compare to fluorescence?
Don't know yet.
Okay.
We're still developing the reactions. They should be, based on everything from the literature, as or way more sensitive.
And last question, I think, before we move on to Dr. Benk, from Felipe Quiroz. Amazing body of work. Your work makes me think of homologous recombination and situations in which the cell has to oversee hybridization reactions. Have you explored proteins that bind to ssDNA to control recombination reactions?
That is an awesome question. I had a graduate student who worked and produced one paper on that, where we essentially used Rec-A to modify in vitro the hybridization logic of invasion. Of course, recombinase polymerase assay is an example of how you harness such proteins to better manipulate hybridization. It's an open area. I strongly encourage you to work in this, or others to work in this. It's really underexplored how to manipulate hybridization logic with proteins in vitro. We did a little of it. Some other folks have done some. The RPA assay is a great example of that, but it's pretty much wide open in my view.
Well, I think with that, we should